Well, good morning, AP Bio students. Mrs. Shelton here, just giving you a Screencastify for Chapter 5, Membrane Transport and Cell Signaling. We'll go over the You Must Knows during our Zoom meeting today, but I also wanted to make sure that I'm putting up Screencastifys for you. So if you want to listen and get more details on some of the content later on, you have the ability to do that. With Chapter 5, couple other things to make sure that you are recognizing with Chapter 5 is that not only do you have your textbook as a resource for Chapter 5, you also have the test prep review series to look over. So that is topic 2, page 50 through 54, so only 4 pages that kind of summarizes Chapter 5. So that's your test prep review series, topic 2, page 50 to 54. And then also, I highly, highly, highly suggest not only do you do the practice questions at the end of the chapter, but use the old textbook study guide, chapter 7. There's a lot of really good practice problems of predicting which way molecules will move. So that is something that you just need to become familiar with. So I highly suggest you do the practice problems for that. So let's switch to the PowerPoint and we'll go over, and I'll just go through all the content that's on your notes so you feel comfortable with that content if you're not understanding what was in the textbook. So let's get this going. Hold on. We'll get this. There we go. Look at this crazy, crazy picture here. So this is actually a computer-generated model of the plasma membrane. And that's really what we're talking about in chapter 5 is we're talking about the plasma membrane. And in looking at this, it kind of looks like it's three-dimensional imagery here, or like a magic eye, if you've ever seen those from the 80s or 90s. If you haven't, come to E7. I've got a couple of them in there. But what this is trying to show you is just all of the molecules that are actually involved and just how complex the plasma membrane actually is. What we're seeing here is the lipid bilayer. So what we have in a lipid bilayer is I have my phospholipids, and that's what I'm seeing over here is phospholipids with their fatty acid tails. Remember, fatty acids are nonpolar and they repel against water, but phospholipids then also have that phosphate head that now gives them a polar region. These are molecules that are amphipathic. Now, what I also have here is I also have integral proteins that are part of the plasma membrane too, because just this lipid bilayer alone, it is semi-permeable. Not a whole lot of things can actually pass through this lipid bilayer. What we're seeing here is just tons of water molecules. These are water molecules on the outside, water molecules on the inside, and we're also seeing that water is coming through this pore here. This might be an aquaporin, might be this protein channel allowing some water to come through. But this is what we need to talk about is this overall structure and then what molecules can pass through and then predicting movement of molecules then through facilitated diffusion or through simple diffusion or even through osmosis. So this is a diagram that you should expect to see on your test. So on your test, you need to make sure that you are identifying all of these parts that we're seeing and all the molecules that do make up the plasma membrane. So with this plasma membrane, like I said, it's selectively permeable, meaning that some stuff gets through, some stuff doesn't. The major component of the plasma membrane is a phospholipid. And so we're seeing a single phospholipid here with its polar head and its nonpolar tails. So these are 2 fatty acids. And then here's my glycerol phosphate that's polar. And because these tails are nonpolar, they repel against water. So they're going to always be turning inward. And then the heads that do like water that are hydrophilic, these are going to be exposed to the water that is inside the cell or also outside the cell. But that's the primary structure then of the plasma membrane is this phospholipid bilayer. Now, what I'm also seeing in the lipid bilayer is I'm seeing these proteins. These are referred to as integral proteins. Now, integral proteins have lots of different jobs, but the one thing I do need you guys to know right now with an integral protein, because it extends all the way through the plasma membrane, this has to be a protein that is amphipathic. It is both nonpolar in the middle and polar on the outside. Now, some of these are serving for cell communication, some of them are serving for ion channels, some of them are transport proteins. There's a whole bunch of integral protein purposes that are part of the plasma membrane. Now, what I'm also seeing in here is cholesterol. So when I see cholesterol embedded into the plasma membrane, I automatically know that this is an animal cell plasma membrane. The purpose of the cholesterol is to help maintain fluidity of the plasma membrane. The membrane has to be able to move. It can't solidify. If it solidifies, then things can't get in and out of the cell. We can't have oxygen and carbon dioxide and water being able to move through the membrane easily. So we need to make sure that we're maintaining this fluidity because, again, the membrane is fluid and needs to be moving. It actually drifts laterally throughout the cell, kind of like on a bubble. If you look at kind of the streaks that come on a bubble and you see a bubble moving, it still maintains shape, but it's moving laterally. The phospholipid bilayer is moving about the cell a little bit and needs to be fluid. So cholesterol is going to be embedded into animal cells in order to maintain fluidity. Now, in a plant cell, what plant cells are going to do is they're going to increase fluidity by putting unsaturated fatty acids in the phospholipids, and that's going to help stop it from solidifying. Now, some other things that you're also seeing in here is I have these glycolipids. So this is carbohydrates attached to fatty acids. I also have glycoproteins. So here's a protein with some carbohydrates attached. These are all involved in cell recognition and cell signaling. Cell-to-cell communication is a part of this chapter that we will intro, but then we'll also talk about cell-to-cell communication with some more specifics when we get to our immune system, talking about that, or when we also talk about the action of hormones, when we talk about our endocrine system. Now, here's my extracellular matrix. My extracellular matrix on the outside, these are proteins that were produced by the cell, so then pushed out from the Golgi apparatus through an exocytosis. And then what you're also seeing is I have these peripheral proteins here. These peripheral proteins, these are not amphipathic proteins. These are clearly polar proteins, but they're here to serve as anchors. So inside, a lot of times these cytoskeletons will actually anchor on here, or also we have secondary messengers that relay signals then to inside the cell when we're talking cell communication. Main parts to pay attention here, though, is the phospholipid bilayer, understanding these proteins, and then understanding cholesterol, and recognize I also have carbohydrates with the lipids, with the proteins. This is also why we describe a plasma membrane as the fluid mosaic model. It's fluid because this moves, and it is mosaic because there are 3 different macromolecules involved in producing a functional plasma membrane. Here's just a closer picture then of the phospholipid bilayer, just so we understand its basic structure. I have 2 layers of phospholipids, and they line up tail to tail. This is my hydrophobic region in here. My fatty acid tails, being nonpolar, repel against water. So even if I came in and mixed up the plasma membrane a little bit and disrupted it, it's naturally going to turn tails in order to hide from the water. The head region then, remember that's the glycerol with that phosphate group, that charge now makes it hydrophilic, liking water. So I end up having this lipid bilayer that forms all membranes, all plasma membranes, all membranes in organelles, in subcellular components, etc. Now, when we do talk about membranes being fluid, and membranes fuse together, this is really important when we start talking about exocytosis, endocytosis, when we talked in the endomembranous system about our transport vesicles going from the rough ER to the Golgi apparatus. Membranes fuse together. So this is just kind of an example of an experiment showing mouse cell, human cell, and merging the plasma membranes together, and now you've got a mix of those proteins. But this is an important way for transport in the cell, out of the cell, or even throughout the cell is through vesicles fusing together. Now, here's just another picture showing you the difference between plant cells versus animal cells. in order to maintain fluidity. And again, maintaining fluidity is really important. So the plasma membrane can still have proper diffusion and movement of molecules through. So this, when I see these kinks in the tails, these are unsaturated fatty acids. And remember, with an unsaturated fatty acids, it's one or more double bonds that causes this bend in this fatty acid tail. So this ends up being a cell membrane that is going to remain fluid even at really low temperatures. And it's important that plants do that. They don't have internal body temperature control like an animal does. So by having more unsaturated fatty acid tails, that's going to maintain fluidity in low temperatures for plants. For animals, animals are going to embed cholesterol. So cholesterol, remember, cholesterol is a steroid. So it is a lipid. So it's also nonpolar. This is why it actually embeds itself within the plasma membrane, because it is a nonpolar molecule and dissolves, again, stays with its buddies here of all of the saturated fatty acids that are here. And this is what animal cells do in order to maintain fluidity of the plasma membrane too. Now, what can actually pass through the plasma membrane? We need to identify and understand with the plasma membrane what can actually move through here is actually lipids. So anything that is nonpolar. So if I have hormones that are steroids, that are lipid based, they can actually just pass right through. But most of my my polar molecules, most of my polar molecules are not going to be able to pass through. They're going to come through, the heads are going to say, woohoo, like dissolves like, come on through. And then those polar molecules are going to hit these non-polar hydrophobic tails and get pushed back out, is what's going to happen. So very, very small polar molecules can get through. And when I say very, very small polar molecules can get through, really just oxygen, carbon dioxide. Those are the two main ones that can simply diffuse through any plasma membrane. Now, water actually gets repelled from a lot of the plasma membrane, but the sheer number of water molecules that are on the inside and outside just bombarding this plasma membrane, some get through. But this is also why some cells, when water needs to just clearly move and freely move in and out, we end up having channel proteins that are called aquaporins that are specifically allowing water to move through. Now, this isn't an aquaporin. This is just an integral protein showing you how proteins can become embedded. And this is going to allow then for these other polar molecules or even transport proteins that need to be moving things in and out of the cell. Now, these channel proteins are specific. So this could be a chloride ion channel. This could be a sodium potassium pump. We're not sure. It could be an aquaporin. But when we now have molecules that can't freely pass, there has to be these integral proteins that are allowing molecules to move in and out of the cell. Again, leading to that concept that plasma membranes are selectively permeable. Here's just a slide that's helping you to see all the different jobs of these integral proteins. We have transport proteins. We have some proteins that actually serve as enzymes for cell recognition, for cell-to-cell communication, for joining cells together, for attachment of extracellular matrix. So integral proteins aren't just for transport, but that's really what we're going to focus on in this chapter are the transport proteins that are allowing molecules to either diffuse or actively transporting molecules into a higher concentration. Now, again, with that concept of plasma membranes fusing together, when we talked about the endomembranous system from the rough ER, now I've got these membranes that are budding off in order to transport my transport vesicles with proteins in them to now fuse with the Golgi apparatus for protein modification. And now I've got another transport vesicle. Well, this is actually where a lot of those membrane proteins are actually produced. I mean, this is where they are all produced, is through this endomembranous system. And now when this vesicle is going to have some embedded proteins, some integral proteins, some proteins that are for export. When this transport vesicle fuses with the outer plasma membrane, well, it now becomes plasma membrane, is what it ends up doing. So when we talk about how does the plasma membrane even get produced, it gets produced by the endomembranous system. So just like proteins, period, we also have the production of more plasma membrane through the endomembranous system too. So some terms that we need to make sure that we are understanding. We need to make sure that we are understanding active versus passive transport. So first of all, if I'm talking about active transport, in active transport, first of all, it's active, so energy is required. So I'm going to be moving things against the concentration gradient, and ATP is going to be required. Most of what we're going to be addressing and the problems that you're going to solve is you are going to be looking at passive transport, and most specifically, it's diffusion. When we talk about diffusion, diffusion is the movement of molecules from a higher concentration to a lower concentration. Now, one thing I need you guys to know is that molecules always want to move from their area of high concentration to their area of low concentration. So here, if I have a semi-permeable membrane that has some molecules, over here is just pure water, or these molecules in high concentration over here, if allowed, will diffuse to then have an equilibrium, an equal concentration of those molecules on both sides of that permeable membrane. So same thing here if we're talking about two different molecules. Again, remember I told you that with diffusion, each molecule wants to move from areas of their high concentration to areas of their low concentration. So when we're solving problems with multiple molecules in solution, we always have to look molecule by molecule, where is their higher concentration, and know that those molecules will be moving to their lower concentration. And that's what we're seeing here with two solutes and diffusion. You will see lots of problems like this, what I commonly refer to as YouTube problems. And with these YouTube problems, what you will be asked to read about is understanding if I have this YouTube with a selectively permeable membrane separating. The problem is always going to tell you what is this membrane permeable to. And in this case, it's only permeable to water, but not permeable to sugar. Well, this is where we now talk about osmosis, where it's just water diffusing. Water is the great equalizer, and water wants to make sure that there is equal solute concentrations on either side of any membrane. So over here, I have a low solute concentration. Over here, I have a high solute concentration. So if sugar can't move, but water can, water is going to move to actually dilute this side over here, making both sides ending up the same concentration. So this is where osmosis happens. Water, and what we're going to talk about is this is a hypotonic solution. Hypotonic means that there is low amount of solute compared to a hypertonic solution. And in my hypertonic solution, hyper means high amount of solute. Water always moves from hypo to hyper. And I'm going to repeat that a lot. We actually look at here's my hypotonic solution, here's my hypertonic solution. Water will always move from hypo to hypertonic in order to balance out my solute concentrations. And that's osmosis. It's just a special type of diffusion. It's a diffusion of water, is all osmosis is. When we do talk about osmosis, this is actually really important for where cells are surrounding themselves and the types of solutions that surround our cells. Now, there's a difference in animal cells versus plant cells. Biggest difference here is that plant cells, each plant cell has a cell wall around it. And so this gives a support in order to prevent a cell from taking in too much water. Now, here are 3 red blood cells, and we're talking about red blood cells. Remember, a red blood cell has dissolved solute in it. It's got hemoglobin, oxygen, some other enzymes that are in there. So there is dissolved solute in all cells. So what our body wants to do as animals is we try to maintain isotonic solutions. This term isotonic means that the solute concentration inside and outside is equal. So if I have an animal cell in an isotonic solution, well, that means that there is a net flow of water that is balanced. I do have water that diffuses in, I have water that diffuses out, but the rate of in versus out is equal, so there is no net change in the amount of water content inside the cell. So this would be my isotonic solution where I have even flow. Now, in an animal cell, isotonic solutions are the ideal solutions to surround our body cells. In a plant cell, however, isotonic solutions is not okay because plant cells need a little additional support. So, what they need to be surrounded by is a hypotonic solution. So, in a hypotonic solution, that means that it's more water than dissolved solute. Remember, water is always going to move from hypo into hyper. These cells both are hypertonic compared to hypotonic. So, this is why we put a lot of pure water with plants. So with this, water is going to flow in, filling this large central vacuole, and that is going to start applying this turgor pressure against the walls of the cell in order to now make this a stronger cell and give it more support. So that is a term that you need to understand is turgid means that there's internal pressure pushing against the cell wall, and that is from this intake of water from osmosis because the plant was surrounded by a hypotonic solution. Now, if we put your cells in a hypotonic solution, this would be bad because water is going to flow from hypo into hyper. Well, we don't have cell walls to protect from this rupturing of the plasma membrane. So when that happens, so like if I decided, that we're not going to do a blood transfusion or, oh, you're dehydrated. I know, I'm going to give you an IV of pure water. Well, you do that into your blood plasma, you've just created a hypotonic solution. And so what's going to happen is your red blood cells are going to take in too much water, causing then the plasma membrane to rupture or to lyse. Because remember, lyse means to cut in science. So that would be in animal cells, this lysing in hypotonic solutions. Now, in hypertonic solutions are bad for both plants and animals. Hypertonic solutions would be like salt water for us. So if we were trying to drink, we're stranded out on the ocean, we're drinking salt water, or you now over fertilize your plants. These would be hypertonic solutions. And what this does, remember, water always moves from hypo to hyper. So this cell actually was hypotonic. It had less solute compared to the saltwater solution you put it in. So this is going to cause water to leave the cell and the cell is going to shrivel. Well, same thing is going to happen in a plant cell. The cell wall is going to remain intact, but as water leaves, well, now the plasma membrane pulls away from the cell membrane because now I've lost all this water in this large central vacuole. And so we call this plasmolyzed when this happens. So sometimes a plant cell can recover from this, but once the plasma membrane pulls away, or now we've got all of the membranes all sticking to each other, even adding water doesn't allow it to recover. Same thing here. So definitely know these terms, hypo, iso, hypertonic. These are relative terms. Just like here, hypo means low, hyper means high solute concentration in comparison. So even though this cell here was isotonic to the isotonic solution, here the cell was hypertonic to the hypotonic solution. Here the cell was hypotonic to this hypertonic solution. So these are all just relative comparison terms of one concentration compared to another concentration. Now, if I have something like an amoeba that is a single-celled organism, it has dissolved solutes and enzymes inside the cell, but these live in freshwater environments. So for this little amoeba to be able to survive, because water is going to start flowing in, it's hypertonic compared to the surrounding hypotonic solution. Well, they have special structures. Like here, this is called a contractile vacuole. And this contractile vacuole is actually going to be collecting excess water that flows into the cell and then squirting it out and pushing it out, actively transporting any water that comes in back out in order to try to maintain the constant pressure or the pressure that it does need to have and not too much pressure to lyse the cell. So when I'm looking at these pictures here, some of the things I need you to be aware of when you see pictures like this, where you see a channel protein or you see a carrier protein, you need to recognize that these are examples of facilitated diffusion. if we're talking about just basic diffusion, diffusion is movement of molecules from high concentration to low concentration. No energy required in order to get molecules to move from their high concentration to their low concentration. But like I said, the plasma membrane is selectively permeable. And being selectively permeable, charged particles can't get through. Large polar molecules can't get through. But sometimes we need them to be able to just freely diffuse. So this is where we'll either have a carrier protein or a channel protein, and you don't need to know which for what molecules. Just understand that these are integral proteins that will allow for that diffusion of specific protein or specific molecules that couldn't just readily diffuse through the lipid bilayer. This is called facilitated diffusion. To facilitate means they needed help. So they need help by having, I like to call them a little doggy door. So this is just a permanent open doggy door that's allowing these solute particles, this specific solute particle, to just diffuse in and out, depending on what the concentration is. Here's a carrier protein. It's just a different shape. These kind of are like, they look to me like garbage cans that have a little flippy lid on them. So whenever the solute particle actually hits that carrier protein, it now opens up and helps that get through. But it's facilitated diffusion. Again, there is no energy required, and molecules are still moving from their area of high concentration to areas of lower concentration is what's happening here. So this could be a gated channel, it could be an ion channel. If it's an ion channel, it just means it's allowing ions through. If it's gated, that means that the particle has to land on top of it. The ligand is what has to land on it, or the solute particle, in order to open it up, or a signal molecule. But just kind of understand those are all facilitated diffusion. Now, when we talk about active transport, in active transport is now moving molecules against their concentration gradient. We are moving molecules into a more crowded space. And in order to force molecules to do that, to go to where there's already a lot of that particular solute, well, we have to put energy into it. We have to force those molecules into that higher concentration area. So this is an example of a sodium-potassium pump. Now, this sodium-potassium pump, super, super, super important for how neurons send signals. And neurons send signals by creating a concentration gradient. So they actually will polarize by putting more sodium outside of the neuron than inside. Now, they actually put sodium outside, potassium inside, and what we end up having is an overall electrochemical gradient of a positive charge on the outside, negative on the inside. More information that you need to understand. But if you hear that term electrochemical gradient, we're now talking about ion distribution. The ions are giving an uneven charge on the outside of the membrane versus inside the membrane. So this is a sodium-potassium pump, and it requires ATP. It's putting a high concentration of sodium outside and creating a high concentration of these potassium ions inside. So it actually does a 3-to-2 ratio. So it pumps 3 sodiums out and then grabs 2 potassiums and pumps those in. But this is an example of an active transport. Now, here's just a summary of what we need to make sure that that you are understanding what we just talked about. Passive transport, molecules are always moving from high concentration to low concentration. No energy is required. Understanding that there's basic diffusion, where those molecules can just move through the selectively permeable membrane or right through the lipid bilayer. That would be our basic diffusion. And remember, osmosis is just a special type of diffusion. It's just diffusion of water. Now, facilitated diffusion then requires the assistance of a doggy door, some type of protein that those specific molecules can diffuse through. So don't worry of whether it's a channel or if it's carrier, it's still facilitated diffusion. But then when we talk about active transport now is moving molecules against their concentration gradient. And so in this case, we're moving molecules already into a high concentration area, and this requires energy in the form of ATP. This is something we're going to talk about a lot in photosynthesis and in cellular respiration, is that your cells purposely will actually create these high concentration gradients of protons. So here's a proton pump. This is an active transport where these little protons, these hydrogen ions, same thing, are going to be pumped into a space in order to create this high concentration gradient. Because we can actually use this high concentration gradient to now diffuse through another protein in order to actually power it, kind of like a dam, how a hydroelectric power plant works. We build up a high concentration of water only to release that water to now serve another purpose for us. So this is an example of co-transport. So in co-transport, this is the use of the proton pump in order to power then a sucrose co-transporter. So sucrose being a disaccharide, this is a pretty big molecule. And if I now have to push this into a cell that now already has a high concentration, so not only do I have a big molecule, but now I want to push it into a high concentration area, well, I need energy. And so sometimes what the cell will do is use ATP to create the high concentration gradient, but then in order to power this co-transporter, now when I have these charged protons diffusing back through, well, that gives enough power to this co-transporter to now move sucrose in. So that's my example of co-transport and setting up this proton gradient or electrochemical gradient in order to then power other activities in the cell. You're going to see this diffusion of protons. You're going to see this in ATP synthase in both photosynthesis and cellular respiration is where you're going to see this. But we have a different type of proton pump that doesn't use ATP. It actually uses the flow of electrons in those two processes. Now, when we're trying to pull in or kick out large particles, we use exocytosis or endocytosis. These are two pictures of endocytosis. These are active transport. It requires energy in order to have the plasma membrane fold around substances that it needs to bring into the cell. So this, again, being endocytosis, I'm pulling things into the cell. There are special terms, though, phagocytosis and pinocytosis, depending on whether it's a large solid particle. So if I'm pulling in a large solid particle or a bacteria or a virus or something like that, then that is a phagocytosis. Pinocytosis, that would then be fluids. It would be solutions that we're pulling in is what is being drawn into the cell in this endocytosis activity. Now, there's receptor-mediated endocytosis. And what receptor-mediated endocytosis means is that we had to receive a signal molecule for that cell to now start folding its cell membrane around and pulling particles in. Here are just some other examples then of, well, actually, we haven't talked about this. So one, if this is endocytosis, just recognize then what that term exocytosis would mean. In exocytosis, we saw that with the Golgi apparatus creating plasma membrane. So now it's the vesicles from inside the cell that are now fusing with the plasma membrane in order to push proteins out or to enlarge the plasma membrane. That would be my exocytosis. Exo for exit, endo for into is what we're talking about. So the very last concept we need to address is we need to address cell signaling and cell communication. Now, again, we're just going to get an overview of it in this chapter. We're going to get specifics in other chapters as we move on. But why cells need to communicate with each other, lots and lots of different reasons, but cells want to signal each other locally. This actually happens in your immune system too. If a cell gets invaded by a virus, they're actually going to release a chemical to tell all the other surrounding cells, like, lock down, I got strange danger in the house. So don't let anything in right now. I have my neurons, they signal target cells, or neurons communicate with other neurons. I also have hormones that travel through my bloodstream for long distance signaling in order to elicit responses in other cells. So all of these, when we actually have signals and how cells communicate with each other, there's always 3 steps involved in cell signaling. So first, you have to receive the signal. So reception is always the first step, which usually it's an integral protein that is the receptor protein on a cell. So if I release this signal molecule, the only cells that are going to end up responding to this signal are cells that actually have this specific receptor protein. So not every cell in your body is going to respond to the same signal. It has to be a target cell or one that has a matching receptor protein in it. So when that signal molecule, so this particular signal molecule actually is a polar molecule that can't diffuse through, so it gets received on the outside, kind of like ringing a doorbell. On the outside of the cell is where the signal is received, but then through the ringing of the doorbell, now this causes inside the cell this transduction reaction. This is just a chemical relay. So this is it initiates this biochemical process that's multi-step that actually amplifies the signal. And we don't need to know the details of this transduction activity. Just know that its purpose is to amplify the signal and then ultimately elicit a response. Typically, the response is to start transcription or affect protein production in some way, shape, or form, is what the response is. But the 3 steps, I have to receive the signal. Then I've got this relay of chemical activities that happen inside the cell. This is my transduction. And then that initiates a response, is what ends up happening. Here's just some different examples of different types of reception. So I can have this G-coupled reaction where sometimes the signal molecule is here and that activates GTP and that activates another enzyme. So it could be a 2-step process for reception. I can also have these ligand-gated channels. And so here, this ligand-gated channel, a ligand is a signal molecule that gets received by this protein. Well, now what happens is that opens it up, ions then flow in, and then that initiates then the transduction and cellular response. This is actually what happens at the neuron. If a neurotransmitter is the ligand, it actually opens up these ion gates in order to allow for that cell response. This is actually showing you the difference in a signal molecule that is a hormone. If it's lipid soluble, remember, since it's lipid soluble, it doesn't have to have an internal or an integral protein in order to receive the information. Hormones can actually go right through the plasma membrane, but they still need a receptor protein. That receptor protein could be out in the cytosol, or that receptor protein could also be inside the nucleus too. But it still has to match up to the receptor protein in order to then initiate transduction and then also then initiate the response. And here we're seeing that the response is initiate transcription, to copy that DNA into messenger RNA in order to start protein production. Here's just another example. Don't freak out when you hear phosphorylation cascade. This is just that transduction, just kind of showing you this series of activity that could actually happen from receiving the signal to response. There's a whole bunch of things that happen in between that amplifies the signal. And this also allows for differences where you can have the same signal molecule that affects different types of cells, even though it's the same signal molecule, same receptor. But if I have a different transduction reaction, then I can actually have a different response in a cell too. So again, here's just another diagram that's just showing you this relay and how this relay of reception, transduction, and then cellular response, those are the terms I need you to know right now. I need you to know that in any cell signaling, there's going to be reception of a signal, transduction, and then I'm going to have a response. And typically the response is initiation of transcription is what's going to happen. So here you're seeing it again. And that, my friends, wraps up chapter 5 in detail. So you can go back through, fast forward, slow me down, speed me up, in order to get the basics then of chapter 5, which is all about the cell membrane. So with that being said, I hope this helps. Let me get off of here and I'm going to stop sharing, and I will see you guys in class to answer any other questions that you may have.